Hello everybody, this is Aloysius Guy with you in a new tutorial. In today's video, we will cover and explore the RNA translation and how protein is synthesized. Okay, so please, before we start, check the time code in the description below to go directly to the chapter that you are interested in. So proteins comes in all different functions within our cells, and also they come in so many different shapes and folding structures, as we can see here. Okay, and these thousands of different proteins that made our body is responsible of showing our phenotype, our physical structures. So the DNA is responsible of showing our genotype, and then the proteins are responsible of showing our phenotype, which are the physical structures. Okay, so the functions of proteins is very diverse. Okay, they are involved in structural support of cells. They're catalyzing some enzymatic reactions, and they have an enzymatic activity. They are also important for cellular support and regulating the transport between cells. And also they are important for immune response. They are an integral part of the immune system. They are very important for metabolic pathways, and they are very essential for regulating cellular defense and repair. Other functions include cellular communications, so how the cell-cell communication occurs. Proteins are required for that, and they are very important for regulating the gene expression itself. So for example, they can induce the expression of some genes, and they can inhibit the expression of some genes. And these functions are just a glimpse of the very diverse functions of proteins. So the question is, if these proteins are very important to our cells and to their structural and functional roles, how proteins are synthesized, and that's what we're going to cover in this video. So in this diagram, this is a very simple diagram to show the flow of the genetic information from a DNA to become proteins. So as we can see here, the DNA information will be transcribed into an mRNA, and then the mRNA information will be translated into a protein. We covered in a previous video this process, the transcription process, which is converting the DNA information into an mRNA. You can check that video in the link that will appear here. In this tutorial, we will cover translation, and we will see how mRNA can be translated into proteins. So just I want to highlight that if you look at the mRNA here, the mRNA is made up of nucleotides, and every 3 nucleotides is called a codon. Okay, so this is one codon, a second codon, a third codon. And when it comes to proteins, so proteins are made up of amino acids. So all these abbreviations are abbreviations of amino acids, names for amino acids. And every codon is corresponding with a specific amino acid. So we will see now how this specificity works in translation. So before we start explaining how the protein synthesis process is working, we need to understand first what is amino acids. Okay, so amino acids, which are the building blocks of proteins. So what is an amino acid? So first of all, amino acids are organic compounds, okay, and all amino acids share this structure, okay, which is having a central carbon atom, which is called an alpha carbon, okay, and this carbon is bound to 4 different groups. So this the first group is the amino group, then the carboxyl group, then the hydrogen atom group, and then there is another group here which is called the side chain, or the R group. So the R group is the only group that distinguishes one amino acid to another. So this structure is shared by all amino acids, however the difference will be in the R group. So different R groups will give us different amino acids. So these are all the amino acids found within the human body. Okay, so in the human body there are 20 different amino acids. All these abbreviations are abbreviations of names for amino acids. Okay, also we can see that for every amino acid there is specific number of codons that correspond to that amino acid. So in the human body there are 61 codons corresponding to amino acids, okay, and 3 codons that are corresponding with what we call a stop codon. So we have UAA, UAG, and UGA. These codons are called stop codon, and we will understand in translation why they are called stop codons. And there is one codon here, it's called the start codon, okay? And also we will understand in translation why is it called that name. So in total, the human body has 64 codons, okay? As we can see, sometimes there is some redundancy. So the same amino acid, it's corresponding to 6 different codons, okay? And this is called some redundancy, which means that the codon that are corresponding with this amino acids can tolerate some mistakes within their sequence. And even so, for example, if we have this sequence and then there was a mistake in the transcription and we get that sequence, they're still giving corresponding to the same amino acid. However, sometimes we can see that some amino acids are only corresponding with just one codon, and these amino acids they do not tolerate any change in the codon sequence. Okay, so that's in general what are amino acids that are the building blocks for protein. So now let's start with translation, which is the process of protein synthesis. So the first thing, what are the main components of translation? So the first component is the presence of a messenger RNA or an mRNA. So we need an mRNA at first. Then the presence of a tRNA or the transfer RNA, which also a key component to translation. Then we have the ribosomes, which are also a key component of translation, and of course the presence of an amino acid. Okay, we explained in more depth the role about tRNAs and ribosomes in a previous video. You can find the link of that video here, so you can click on it and you can gain more information about that. But today we will focus on their role in translation, and we will see how they are very important. So the first step of translation, it's called initiation. Okay, so initiation is the first stage of translation, and there are 2 main parts of initiation. So I call them 1A and 1B. Okay, so 1A of initiation, it will be tRNA binding to amino acids. So within the nucleus, tRNAs are transcribed, and once they are processed and they become mature tRNAs, they move out of the nucleus into the cytoplasm. Okay, once tRNA are in the cytoplasm, they will have a very specific structure. Okay, and within that area of the tRNA, they will have a very specific sequence of nucleotides, which is, as we know now, they are called codons. Every tRNA has a specific sequence of nucleotides at that site. Okay, if you want to know more information about the structure of the tRNA, just click on the video that will appear here and you can expand your knowledge of that. So this specific codon sequence corresponds and determines the specific amino acid that binds that tRNA. Okay, also this specific sequence is complementary to a specific sequence on the mRNA. Okay, and we will see later during the elongation and also the second step of initiation how this specificity is important for binding to mRNA. Okay, so yeah, so once tRNA is in the cytoplasm, they bind a type of enzymes. They are called amino acyl tRNA synthetases. Okay, and these enzymes are very important because they provide an active site to join every tRNA with its corresponding amino acid. So for example, AUA, this sequence or this codon corresponds to this amino acid. Therefore, this enzyme will only join tRNAs with that codon to this amino acid. Okay? And since we have 20 different amino acids, there are also a corresponding 20 different synthetases. Okay, therefore synthetases are very important in joining the right amino acid to the right tRNA, which carries the right codon. So if that sequence has changed, then this tRNA will not be able to bind to this tRNA that binds isoleucine. they will bind to a different synthetase that binds a different amino acid. So that's why this process is very important. The second step of this process is using ATP or using energy in the form of ATP to link the tRNA to its corresponding amino acid. And this link occurs through formation of a covalent bond between the amino acid and the corresponding tRNA. And if we can see here, the tRNA has a much longer arm on one side, which is a single-stranded arm. And this arm is called the acceptor arm. It's a single-stranded arm that binds the amino acid. Okay. Once tRNA is bound to the amino acid, they are dissociated from the synthetase, okay? And in that form, the tRNA is a charged tRNA that is ready to bind to the ribosome and to the mRNA and start the translation process, okay? And also, I want to just re-emphasize the importance of this information, which is that synthetases really governs the binding of specific tRNA with specific anticodons, and that ultimately governs the binding of the corresponding amino acid. So now we will cover the second part of initiation, or 1B, which is the ribosomes binding to mRNAs. So within the nucleus, there's something called the nucleolus. So within nucleolus, ribosomal RNAs get incorporated into the large subunit and small subunit. If you want more information on that, just check that video that will appear here, and you will get more information. So once these subunits are formed, they move out of the nucleus into the cytoplasm. So what happens first is that these small subunits will start looking for mRNAs. Once they identify mRNAs, with the help of proteins called initiator factors, small subunits can bind to this 5 cap of the mRNA. And that is the start of translation. So what happens is that this small subunit will start moving through the mRNA, okay, in the direction from the 5' to the 3', and until they find a very specific sequence, which is, we call it a start codon. So if you remember the table that I showed you earlier, there is start codons and stop codons. So here is the function of the start codon. So its main function is that it needs to be identified by the small subunits, and once they identify it, they attach to it, and then now the sequence is ready for the binding of the first charged tRNA that has the anticodon complementary to that sequence in specific. So as the tRNA binds to the sequence, which is the start codon, what happens is that the large subunit of the ribosome will also join. So these two things happens very simultaneously. Okay, so the tRNA will just bind to the complementary sequence, and then the large subunit will follow. Okay, and that site, which contains the start codon, is called the P site. Okay, and now what we have is a complete ribosome mRNA complex, and by that the initiation step is finished, and now we will start the second step, which is the elongation. Okay, so elongation is the process of forming polypeptide chains. That's the simplest definition of that stage. Okay, so as we can see here, what we saw before is that the first tRNA bound to the P site where there is the start codon. Okay, so what happens is that then a new tRNA will bind to the next codon downstream of the start codon. This tRNA contains an anticodon that is complementary specifically to the sequence. Okay, so what happens is that now we call this site an A site, which is the site of binding of new tRNAs. Then what happens is that the ribosome will catalyze an enzymatic reaction that forms a peptide bond between these two amino acids. So this amino acid will be dissociated from here, transferred to this tRNA, and then both subunits will move one codon downstream in that direction. So as we can see here, this is the movement of the ribosome. Okay, so once this tRNA has no longer has an amino acid, they will just be moved to the E site, or the exit site. And now this this tRNA will just dissociate from the mRNA, and this is called deacylated tRNA. And now we have a growing polypeptide chain on this tRNA, and also the A site now is free for a new tRNA with an anticodon complementary to this sequence to bind here. Okay, this movement of the ribosome, one codon downstream of the start codon, is called translocation. So now we get to the final stage of protein synthesis or of translation, which is called the termination. Okay, so in termination, what happens is that the both ribosomes will be continuing moving through the mRNA and building this polypeptide chains, and new tRNAs will be just coming there, and the whole process will be carried over and over again. Okay, until the ribosome reaches a sequence called stop codon. Okay, so the special thing about this stop codons, if you remember the table that I showed at the beginning, there are 3 different stop codons. Okay, so these stop codons cannot be bound by any tRNA. Okay, so that's why the ribosome will stop there. Okay, and that's going to be the last tRNA. However, what can identify these stop codon are a number of proteins called release factors. Okay, so what happens is that release factors will bind to the stop codon, and then these release factors will catalyze the cleavage of the polypeptide chain from the last tRNA. So after cleaving the polypeptide, now the polypeptide is dissociated, and this will be followed by the dissociation of both ribosomal subunits, the tRNA, the acylated tRNA, and the release factors. Okay. So what happens after all the components are released from the mRNA and the translation has finished? So the first thing that will happen is that this polypeptide will start undergoing some folding, forming the protein structure. Okay, and I will be doing a tutorial in the future that covers how proteins are folded into very specific shapes. Okay. In terms of the ribosomal subunits, they are free now, and they can just bind to another mRNA and start translation again. For tRNAs, they can be reused again, so they can go to the synthetases, bind to another amino acid, and they just and they're just going to be joining another translation process. And then for the mRNA, it can be also translated more than once. Okay. Before they are degraded and removed. And that's the end of the translation process and the formation of a protein or a polypeptide chain. So here is just a summary of all of the 3 main stages of translation. So initiation, which is the step of binding of small subunit to mRNA, binding of tRNA to the start codon, and then this is followed by the binding of the large subunit. And then the second step, elongation, where the new tRNA binds, the amino acid will be cleaved from the old tRNA, associated or bound to the new tRNA, and then the tRNA will be the old tRNA will be exiting the whole complex. And then the last thing is the termination, where the ribosome reaches a stop codon, and then the release factor bind to that stop codon, cleave the polypeptide chains, and everything will be then dissociated from the mRNA. And here are some exam-like questions that will help you to understand the topic very well. So you can check them and try and have a go at answering them. And with that, guys, we sadly came to the end of this tutorial. I hope it was easy to follow and understand. Please don't forget to like, comment, subscribe, hit the notification button so you're going to be notified of the new videos, which are going to be linked to these videos. So it's very important that you keep watching. And please don't forget also to go to the description so you can just go directly to the chapter that you're interested in. Thanks for watching.